Good evening, dear students. I hope you all are feeling very confident and well prepared for your examination. Welcome to our online revision class. Today, I will try to refresh the key topics and focus on crafting the effective exam answers. First of all, let me go through the important questions of each topic. And then I'll try to say I'll be discussing few topics. That is not just to teach something, instead to say how to write the exam, how to prepare, how to put in your knowledge on the paper. So let me start now. Anyway, at the onset, we should all thank this platform, Eden platform, for giving the opportunity for the revising the topics. Their caring for the students should be appreciated, and hats off to that platform. So let me start now. First of all, let me go through the important questions topic by topic. I'll just start from the chemistry part. In chemistry part, of course, the beginning we always study the definition, biologically important, the classification with example. Long back once it has been asked. Then a few times, epimer anomer was a 3-mark question. Once or twice, homopolysaccharide came as was a 3-mark question. Then a repeated question from chemistry carbohydrate is mucopolysaccharide. See, that's why I written. See, these are some subheadings what we are supposed to know there. Mucopolysaccharide. First of all, definition part. How will you define it? It is, see, we always say this way, it's long, unbranched, heteropolysaccharide. These terms each are important. Long, unbranched, heteropolysaccharide. What is mainly present in the extracellular matrix and consists mainly of uronic acid and amino sugars. Then say its names and what are the components of it, what are the functions of it, in that way it goes. I hope you remember the terms like hyaluronic acid, dermatin sulfate, chondroitin sulfate, keratin sulfate, and what more. Okay, so these are the mucopolysaccharides. And say about its functions. Okay, once or twice this question, we are very familiar at test. What you do in a clinical laboratory, in the laboratory, is Benedict test. That also has been asked. If the Benedict test is asked, you're supposed to say it's to identify the reduced sugar. Say what are the components of Benedict's reagent, that principle of the reaction, and why it is called as semi-quantitative test. So these are supposed to write under the Benedict test. So just trying to say some important points. And next is about the lipids, chemistry of lipids. In that, classification of lipids, of course, is a general question. And other two, second and third questions are very repeated questions of lipids. They are essential fatty acid. Again, how will you define it? And these are some key subheadings for supposed to give for essential fatty acid. Definition like fatty acid which are not synthesized in the body, which should be supplied through the diet. Name are linoleic acid and linolenic acid. Then what are the functions? And deficiency manifestation. It goes like that. Okay. Same way, in amino acid metabolism also we have amino acid chemistry. We have another question, essential amino acid. Same definition. Amino acids which cannot be synthesized in the body, but which should be supplied through the diet is called as essential amino acids. And then its name comes around 8 essential amino acids are there, 2 semi-essential amino acids are there. So study that also. And then in essential amino acid, what will you write along with that? There, more than the functions and deficiency manifestations, should say a protein which contain all the essential amino acids called as complete protein, and which lacks one essential amino acid is called as incomplete protein, and any protein which lacks more than one essential amino acid is called as poor protein. Write the examples for it. Of course, continuously, if the essential amino acid is not taken up by through our diet, then naturally our weight gain will not happen. Immunity may reduce. That is something you can write on essential amino acid. Okay, I'll come back to the topic of lipids. One more question there, phospholipids. That's also a repeated question, phospholipid. As you know, it's mainly present from our cell membrane onwards. And it is amphipathic in nature. So it's very important actually, important biomolecule in our body, because of which it's an important question also. Phospholipid again, you are supposed to write like definition, classification, how will you classify it? Classification is glycophospholipid and sphingophospholipid. And write its functions one by one. Okay, that's about the chemistry of lipids. In chemistry of amino acids and protein, of course, once or twice the classification of protein has come, classification of amino acids come. But the major question, a repeated question from the chemistry of amino acids and protein is structural organization of protein. So I'll take some few minutes to say about that. Of course, plasma protein, it's important topic even for your practical examination, and functions of albumin, its classification, functions of albumin is important. Immunoglobulin, Y-shaped globulin chain, that also is important question. Okay, let me have some few ideas about the structure of protein. Protein structure, as you know, we have always explained that 4 levels: primary structure, secondary, tertiary, and quaternary. And that primary structure is maintained by covalent bonds. All other 3 are maintained by non-covalent bonds. Okay, because of which any denaturing agent can easily break the non-covalent bonds, because of which secondary, tertiary, and quaternary structure that also get destructed during denaturation. Only primary structure can withstand, or primary structure is not altered during the process of denaturations. Okay, what do you mean by primary structure? I'll just give you some very few, some most important points about the primary structure. Of course, what is primary structure? Primary structure, it defines how many amino acids are there in a protein or a peptide chain, and in which order it is arranged. So I can say in this way, number and sequence of amino acids in a protein. Or something is not only protein, just it will be what do you call this? It's a polypeptide chain. And mainly, and what is importance? Okay, it's maintained by, this primary structure is maintained by covalent bond. The major covalent bond is peptide bond. Of course, there are some disulfide bonds also in some polypeptide chain. So these covalent bond maintain the primary structure. I should say that, then say a few points about the peptide bond. It's a bond between a carboxyl group of one amino acid with a amino group of another amino acid. It has a planar nature, just a partial double bond character. It can have a cis or trans nature, mostly it is in trans form. Okay, that points you're supposed to say about peptide bond. And another thing, what is the importance of this primary structure? Primary structure determines the secondary, tertiary, and quaternary structure, and thereby the biological function of a protein. Okay, so function of protein depends on the primary structure. And that's a structure that defines or determine the higher level. That means second structure, what should be the secondary structure? What should be the tertiary structure? That's also determined by the primary structure. And another one is about, okay, what is the significance or feature of primary structure is that it is not altered during the process of denaturation. So if the question is primary structure, write first the definition, then how it is maintained by covalent bonds, say about the covalent bond, then say a few points about peptide bond, write peptide bond as a subheading, and then write few points about peptide bond. Then features or most unique features of primary structure. You can write some examples about insulin in that. Okay, that's about the primary structure. And another question here. So primary structure can be a 3-mark question. And secondary structure is another repeated question. If you understand the concept behind the secondary, you won't forget it. Secondary structure is also about this one. See, a protein, this is about the hemoglobin, in that a globin chain, in that a part of a globin chain, how it looks like. That's called a secondary structure. It can be a helical structure or it can be zigzag. So secondary structure means local shape, simple words, local shape or local conformation. So this can be a part of a peptide chain, how it looks like. Some will be like this, some will be a zigzag, it goes like that, no? So second structure, local conformation means local shape, 3-dimensional structure of a polypeptide chain, a part, not totally, a part of it. Or we say, in a peptide chain, what are the common regular folding pattern or common recurring structures by the stable arrangement of amino acid? Or more specifically, what are the bonds or interactions between the amino acid which are nearby in a linear sequence? That means first amino acid, it might be having a link with the third amino acid or the fourth amino acid. So that interaction is defined here. So this is some first few points you're supposed to write about second structure. Local conformation, recurring folding patterns or regular folding patterns, interaction between amino acids which are nearby. So these first 3 sentences give a concrete idea about the second structure. And mainly we have 2 second structure, alpha helix and beta sheets. Okay. Just we'll go through each. Alpha helix. I'll just try to have a comparison. Alpha helix, you know, it's coiled structure. So because of coiling, it will be packed, tightly packed. And this is coiled structure where the side chain of amino acids will be extending outwards, not to the center. Okay. So we'll just have some important points. I'll just show you. A spiral structure, tightly packed. That means around 3.6 amino acid per turn, or distance between 2 amino acids is around 1.5 angstrom. So whenever you see an alpha helical structure, there'll be only one coil, one spiral or one coil polypeptide backbone. And then they are maintained by, this alpha helical structure is maintained by, of course, hydrogen bonds, and that will be intra-chain. That means what? The first will be having a link with the fourth one. Second will be having the fifth one, like in that way it goes. Okay, so mainly maintained by intra-chain hydrogen bonds, and that will be parallel. That will be, see, these are the intra-chain hydrogen bonds. They will be parallel to this central axis. They will be parallel to the polypeptide backbone. And around in our hemoglobin, around 8 segments, 8 set of alpha helix is there. 80% of hemoglobin is having alpha helical structure. And our keratin protein also contain alpha helical structure. So this is some points about alpha helical structure. If you understood this part, then beta sheets you can say. Beta sheets is zigzag. Here it is in alpha, this is spiral structure. Beta sheets zigzag in nature. Alpha is because of coiling, it is tightly packed, but here it's loosely. Okay, and distance is more. And then in one beta head sheets, you can see more than one peptide, polypeptide chain. I'll just show you again. This is about our beta zigzag helix. So appears as beta sheet zigzag, fully extended. There it is 1.5 angstrom, the distance with amino acid. Here it is 3.5 angstrom. And there is only one peptide chain, polypeptide chain. Here you can see 2 or more peptide chains when we say beta sheet. So that's why we always use the word beta sheets. And there it is only intra-chain hydrogen bond. Here it can be intra-chain or inter-chain. There it is the hydrogen bonds was parallel to the peptide bond, peptide backbone. Here it is perpendicular. On one example, carbonic anhydrase. So these two, if you study and compare and study, it will become easy. And then don't stop here. One more point, extra point, which gives you more 0.5 marks extra, is about some superstructures. We have some superstructures. We have some beta turns and loops. A short segment, you can see that. See, here between two things, two types of proton structures can be linked. Short segment of amino acid that join two units of regular secondary structure. It can be beta bend or beta turn, it can be a loop. Okay, so just mention these terms. Short segment that join two regular secondary structures, beta turn and loops. So that also comes under the secondary. So this much points you can write about secondary structure. So understand the idea and write, then you won't forget even. Okay, so this much about our secondary and primary structure. They are always a repeated question. That's why I just said about that. Okay, coming back to the next, again, the important questions. Enzymes, as you knew several times, the classification was asked. Their classification, I'm sorry, based on the type of reaction it helps. We have a word, othylil, just write accordingly, because whenever you say enzyme class 1 is nothing but oxidoreductase. Enzyme class 2 is transferase. Enzyme class 3 is hydrolase. Then lyase, isomerase, ligase. And as you all know, we have one more. Up to maybe 2 years before, we had only 6 classes. Now we have one more class. It's called as translocase. So all the 7 classes, write with examples. Then another question, few times this question has been asked, enzyme specificity. As we know, a unique feature of an enzyme is specificity. And in vitro way is specific. One is called as absolute specific. To act on a specific compound in a specific reaction. For example, glucose oxidase just oxidizes glucose, nothing else, nobody else. So absolute specific or substrate specific. Second specific is group specific. Example is hexokinase. It can phosphorylate glucose, it can phosphorylate galactose, it can phosphorylate mannose, any hexose sugar, hexokinase. So that is group specific. Third is stereo specific, and fourth is bond specific. Bond specific example about pepsin or trypsin can say it will break the peptide bond, but not all the bonds between specific amino acids. Peptide bond formed between specific amino acids alone will be hydrolyzed using pepsin or trypsin. So it's bond specific. We have stereo specific. For example, L-amino acid oxidase. It can oxidize only L-amino acid, L-stereo specific. So in that way, that's one question that has been asked. So write the 4 specificities. I will repeat once again. Absolute specific or substrate specific, group specific, bond specific, stereo specific. Okay. Then active site, as you know, where the substrate bind and the reaction occurs. And then another a repeated essay question is factors affecting enzyme activity. Then we have supposed to draw some graphs, and other things are there. That means effect of temperature, pressure, pH, substrate concentration, enzyme concentration, and product concentration. Then the presence of coenzyme, cofactors, presence of inhibitors, radiations. These all can be added up in that. Factors affecting enzyme activity is an important essay question. And then a repeated question that has been asked is about the competitive inhibition. Inhibition as such is important. Why competitive inhibition is important? Please write this point, you should know. Most of the pharmacological action of drugs, that is medicine, acts by competitive inhibition. So because of which is very important for us. So competitive inhibition, here there's a competition between the inhibitor and the substrate to bind at the active site. It is reversible nature, Km values increase, Vmax remains constant. These are some features of it. And then write the examples. You study, for example, with the enzyme name, That'll give you few marks. Then clinically important enzyme related to liver disease and myocardial infarction. In that nowadays, myocardial infarction, we have some other different biomarkers. So nowadays, we are not specific about that. There's no point in studying the enzyme name alone, the myocardial infarction. So, but this question, liver disease is important. We have some three major enzymes, AST, ALT, and then ALP. Aspartate transaminase, alanine transaminase. These two are elevated in hepatocellular diseases. And acute cases, ALT will be more. And the chronic cases, AST, aspartate transaminase, may be more than ALT. Then in obstetric jaundice, ALP increases. In chronic alcoholics, alcohol abuse, the gamma GT will be elevated. And 5' nucleotidase. So these enzymes you can write in liver diseases. Write its normal level, in which conditions it increase. That's what it's supposed to say. ALP, as you know, ALP elevates mildly or moderately in liver disease, very high in obstructive jaundice, and drastically in bone disease, around 25 times elevation in bone disease. So, and you can write that points also. So mainly you're supposed to add up in that way. Then, okay, another question once or twice asked is isoenzymes. You know what are isoenzymes? Isoenzymes are the enzyme. Iso means same enzyme. That means what? Same action. Then what is different? Their physical properties, chemical properties, or site of origin. I'll always say this way: when you study isoenzymes, study the isoenzymes are the classmates in a class. They all are supposed to be here for the same function. Okay, that means catalyze the same reaction, but they are not from the same place. They are physically not same. Their behaviors, they are not same. Same thing. Isoenzymes are same enzyme or enzyme having the same activity, but differs in physical property, chemical property, or site of origin. Then isoenzymes are short note. You are supposed to write, first you write the definition for it, then write some example. As you know, CPK has 3 isoenzymes, LD has 5 isoenzymes, write the examples. Then say biochemical reason for the isoenzyme formation. Okay, we have 5 reasons are there. Then how we differentiate or how we identify them. What are the mechanisms, methods by which we differentiate them? Or other words, what are the features which differentiate it? Okay, so that much points about isoenzyme. And one more question about Km value. Twice or thrice this question, Km value, and what has been asked. As you know, when you study about the effect of substrate concentration on the enzyme activity, we draw a graph and Vmax by we know what is Vmax, and for the Vmax by 2, there is a substrate concentration that to attain Vmax by 2, half the maximal velocity is called as Km value. You know it. Write the features of it. Okay, and write the Km value is asked. You can add up in this way. What happened to Km value in competitive inhibition, non-competitive, and uncompetitive inhibitions? Okay, that's all about the enzyme topic. Metabolism of carbohydrate. Several times, digestion absorption of carbohydrate has been asked. Remember that this question, we think it's not important, but several times it has been asked. In digestion part, I want to put it this way. Digestion happens one in the mouth using salivary amylase, in the intestine using pancreatic amylase. And how the amylase act? You are supposed to say that salivary amylase and pancreatic amylase both randomly breaks alpha-1,4 glycosidic linkage. There is this point we expect from you. It will not break sequentially, it will be break randomly alpha-1,4 glycosidic linkage in the starch. Okay, or in the other carbohydrates. That's what amylase do. Okay, so to write about this action of amylase and limit the exchange of the product in the mouth, then intestine after its action, disaccharidases like maltase, isomaltase can act. Then oligosaccharidases can act on it, convert to monosaccharides. Then sucrose convert is digested using sucrase enzyme, maltose using maltase, lactose using lactose. And then how it is absorbed. In the intestine is absorbed using as we know, for glucose and galactose, we have S-GLUT1, sodium dependent glucose transporter 1. You are supposed to write that. And then you write as a clinical importance, you write about lactose intolerance. That means you are supposed to write about digestion absorption of carbohydrate. Then a question that everybody is familiar with is glycolysis, has been asked several times. Another important question is about the gluconeogenesis and glycogen metabolism and its regulation. Important question. And important repeat question is HMB pathway, significance of HMB shunt pathway. Another question is regulation of blood glucose, how it is maintained. Then about diabetes. Once it has been asked about diabetes, what is diabetes, how it is maintained. Later on, I will be explaining about gluconeogenesis and HMB pathway. So these are some important questions in carbohydrate metabolism part. Then about metabolism of lipids. Again, here also digestion absorption of lipids. And here you should say about the lipase enzyme, lingual lipase, gastric lipase, and then pancreatic lipase, cholesterol esterase, phospholipase. About the 5 enzymes you're supposed to say. And its action. Of course, you're supposed to say the action about the role of bile salts here in digestion absorption, digestion, lipids, after we consume the fatty compounds, because of its hydrophobic nature, it will be together. For its digestion purpose, it should be converted to small lipid molecules. That is done with the help of bile salt. That process is called as emulsification. So with the help of the process of emulsification, with the help of bile salt, emulsification happens, then the enzyme can act on it. Okay. After that, after the digestion, then for the absorption part, again, bile salt helps by formation of micelles. Okay. So that you're supposed to say. And then a number of times, beta oxidation of fatty acids was an essay. Ketosis has been asked as a 5-mark question. Ketone bodies. Ketosis means what? Ketonemia, ketonuria, and smell of acetone in the breath. It's called as ketosis. Conditions with these all things. Okay. Then another repeated question is about the biologically important compounds produced from cholesterol. Here, see, very easy to remember, cholesterol. Divide that word into chole and sterol. Chole means bile acid and bile salt. Understand that way. So important compound produced from cholesterol are, one is bile salts and bile acid. Then sterol, second word, sterol, steroid hormone. Then third one, everybody knows, when the sunlight falls on 70 head of cholesterol, convert to cholecalciferol. Another name for cholecalciferol, vitamin D. So these are 3 major compounds produced from cholesterol. And one more function for cholesterol, that's one of the major components of our cell membrane, which maintain the fluidity. So, and write about the synthesis steps. That's what we meant. We expect from you on the 4th question. 5th is on the question of lipoprotein. In that, HDL cholesterol has been asked several times. Metabolism of HDL has been asked. It's called as good cholesterol. Transport cholesterol from tissue to liver, thereby helps in the excretion and prevents atherosclerosis. So that part, HDL is important. So that's an important question of lipoprotein. Then last part, atherogenesis and atherosclerosis. That has not been asked so far, but just for sake of completeness, I just added that too also. That's all about the metabolism of lipids. Another one is about the metabolism of amino acids. In that, transamination several times has been asked from the word itself, transamination, transfer of amino group from an alpha amino acid to alpha keto acid to form a new amino acid alpha keto acid. Okay, that is one question. And see, when transaminase is asked, write the definition, subheading. See, we actually, when we correct it, no, digital valuation. So the subheadings and the way which you write is very important. That's why I'm telling about the subheadings. Transaminase, say about the subheadings, write about the heading, sorry, the definition part. Then say the examples about of it, at least two of AST and ALT, as you said, as we just now discussed, which are relevant in hepatocellular diseases, AST, ALT, aspartate transaminase, alanine transaminase with the example you can write. Then significance of transamination. Significance aspects we can say the first step in the amino acid catabolism, whereby non-essential amino acid can be synthesized. That's second point. Third, there's an interconversion between the one amino acid to another. Fourth is one major part. Through transamination, some alpha keto acid is produced that can be used for gluconeogenesis. So transamination provides substrate for glucose synthesis. Very important. Okay, write that point. Clinically important to say about AST, ALT. AST is elevated in MI. Both AST and ALT are elevated in liver diseases. So in that way, the subject is right. Transamination. Then transamination plus oxidative deamination together we call it as transdeamination. That question also can be asked. Another question is about transmethylation. Is about what? Methionine, amino acid methionine, its active form called as S-adenosine methionine. It helps in transmethylation reaction. There are a number of reactions. So what is transmethylation reaction? Transfer of methyl group from S-adenosine methionine to any methyl acceptor. Thereby, this is a process thereby we can synthesize creatine. Process is involved in our epinephrine, norepinephrine synthesis, epinephrine and metanephrine synthesis, choline synthesis, serotonin synthesis. So that's the important. So in that way, a number of biologically important compounds can be produced through transmethylation reaction. So that examples you are supposed to find. One major essay question is urea cycle. How ammonia is disposed from the body. Trying to say about urea cycle. And if sometimes the question can be a direct question like urea cycle. Anyway, if the urea cycle is a direct question, if question comes, that cycle alone will not fetch you the marks. You are supposed to say a definition for it. Why urea cycle? During amino acid metabolism, ammonia is produced. It is neurotoxic. So this ammonia is converted to a less toxic and easily excretable form called as urea. So that's the importance of urea cycle. So that point should be there in your paper. Urea cycle is a process of detoxification of ammonia. This part you should know. Okay, that's an important point you're supposed to add up. Or first point there. After definition, cite and say it's about the detoxification of ammonia. Then say the source of ammonia through from amino acid catabolism. What are the ways? Transdeamination, oxidative deamination, then non-oxidative deamination process, then action of bacteria on the food or bacterial metabolism, then breakdown of purine and pyrimidine also produce ammonia. So these are the source of ammonia. Then urea cycle includes the liver. Okay, so it should be transferred towards liver. Next point is transport of ammonia. After it reaches the liver, then only urea cycle. So urea cycle essay question, urea cycle, start with the definition side, then say the significance of it, then say the source of ammonia, transport of ammonia, then is the word urea cycle, the process. Hope you understood. So for a urea cycle essay, just writing the cycle alone will not give you the full marks on that. You would have studied cycle well, but without writing all other things, you won't get the good marks on it. And then write about the energetics disorders. At least this much points you're supposed to know about the urea cycle. Okay. Another question, a 5-mark question that has been asked is about the glycine. What about glycine? See, what are the major biologically important compounds produced from glycine? The question can be two ways. One, importance of or the uses of glycine. Biologically important of glycine. Another question, biologically important compounds produced from glycine. If the question is biologically important compound produced from glycine, of course, then you can say about our heme synthesis, purine, creatinine, glutathione. And one of the major components of collagen, or major protein collagen, is glycine. So these are the biologically important compounds produced from glycine or using glycine. But if the question is biological importance of glycine, then along with the compounds produced, we're supposed to say other functions of glycine. What are the other functions? One, help in the conjugation, our bile salt, bile acid, cholic acid and cholic acid. They are conjugated using glycine. So it forms glycocholic acid, glycocholic acid to make it more soluble. So conjugation. Then excretion of some compounds. Example, benzoic acid. What is present in the juice and as a preservative and jams. It is there as a preservative for its removal from the body. In other words, for its detoxification process, it should be attached with glycine and form hippuric acid. That's another biological importance. And then another biological importance is it can act as a neurotransmitter. So conjugation function, excretion function, and function as a neurotransmitter. That also should be added. So read the question, understand the question about glycine thoroughly. That's my point. If the question can be either biologically important compounds produced from glycine, then L is the compound, right, in the synthesis. We have mainly 4 compounds and 1 protein. Compounds are purine, heme, glutathione, creatinine. And the 5th is collagen. 1/3 of collagen is glycine. Okay. And if the question is on biological importance of glycine, then along with these 5 important compounds, write about other functions. Very important functions is conjugation function, excretory function, and excretion of the compounds, then neurotransmitter. Okay, I hope you understood. That's clear. Okay. Another question, repeated question about, of course, about our aromatic amino acid. Name aromatic amino acid, write on the metabolism of phenylalanine tyrosine. That has been asked several times. Because from the tyrosine, the major compound produced are catecholamine, thyroid hormone, melanin. This is an important question. So we can study on that also. First, the question is writing about from phenylalanine to tyrosine by phenylalanine hydroxylase enzyme. Then, as I said earlier, for any amino acid, the breakdown process, the first step is transamination. So here again, tyrosine transaminase breakdown. So para-hydroxy phenylpyruvic acid is forms. Then step by step, there comes homogenesic acid, formal acetate, and fumarate and acetic acid. This is the catabolic steps. Then write about the if an essay. Of course, the question will be about not only the when we ask about metabolism, say about the important compound produced also. And of course, enlist the disorders related to it. Of course, you can write about phenylketonuria, alkaptonuria, and you can write about the albinism. Okay. So that much about aromatic amino acids is an important question. Please note here, in this one topic, we have 2 important repeated essay questions. One is urea cycle, other one is metabolism of aromatic amino acid mainly about phenylalanine and tyrosine. So Write about the catabolic pathway, along with that the major compounds. You should know one more thing, that many a times a 5-mark question has been asked, like glycine, biologically important compounds produced from tyrosine. So 2 5-mark questions, important questions are there. Biologically important compounds produced from glycine, and biologically important compounds produced from tyrosine or phenylalanine. Then transamination is a 3-mark question. Transportation 3-mark question. Phenylketonuria, alketonuria has been asked as 3-mark questions. Okay, so very important questions are there. Of course, methionine, that can be a question. See, about the SAM from methionine, X understood methionine production, then transfer reaction, and then homocysteine methionine, or homocysteine to cystathionine. Because we have studied these steps along with folic acid and B12, and along with the PLP also. So an important topic actually, in fact. That's why that I have put that metabolites of sulfur containing amino acid and homocystinuria as a question. Okay, so these are some important repeated questions or important questions on metabolism of amino acid. Excuse me. Another question, another topic, oxidative phosphorylation. Very important one. Most important is components of electron transport chain. See, if the question is about electron transport chain, straight away you say about there are 2 types are there. What are they? Mobile components and fixed components. Fixed components are complex 1, complex 2, complex 3, and complex 4. And mobile components are ubiquinone or coenzyme Q. It's also called like that. Then other one is cytochrome C. So say what are the compounds on it and how the electrons are transported from each. Say the names of that. Complex name, not only just the word complex 1. Understand this way. Complex 1 is the one which helps in the transfer of hydrogen from NADH. So the name NADH dehydrogenase complex. Very easy. Study in that way. Complex 1 help in the removal of hydrogen, dehydrogenation from NADH. NADH dehydrogenase complex. Since contain more than 1 or 2 one compound, we call it as complex. Complex 2 help in the removal of hydrogen from the succinate. So the word succinate dehydrogenase complex. Complex 3, very easy. That's the one which gives electrons towards cytochrome. So it's called as cytochrome reductase. Those who provides electrons, we call this reduction. It's a reduction process. So complex 3 help in the reduction of cytochrome. So the word cytochrome reductase. Complex 4 gets electron from cytochrome. So it is oxidizing the cytochrome. So the word cytochrome oxidase. Very easy. Study in that way. 4 complexes name. Instead of just writing complex 1, complex 2, complex 3, complex 4, we won't give you the marks for that. Instead, you are specifically supposed to write the names of it. Names you study in this way. NADH dehydrogenase complex, succinate dehydrogenase complex, cytochrome reductase, cytochrome oxidase. Say what are the compounds in that and say about the electron transport. Schematic representation on electron transport through that. Okay. Then say about the complex 5 also. It's also called as AB base. A 3-mark question that is about the Kenny osmotic theory. Electrons, when are transported through these complexes, the protons are pumped out from the intermembrane space from the mitochondrial matrix to intermembrane space. I'm sorry, I'll repeat once again. When the electrons are transported through the complexes, protons from the mitochondrial matrix is pumped towards the intermembrane space. Okay, and this will produce a potential difference, PMF, proton motive force, or electrochemical gradient, by which the protons, again, that protons, other words, H plus, are pumped back to the matrix through the complex 5, which results in the ATP synthesis. That's about the chemi-osmotic theory. Okay, schematically you can say about that. Then for the last our additional batch examination, this was a question: high energy compounds. Any compound which can produce around during catabolism, produce more than 7.3 kilocalories. That's the energy of an ATP. That's called as high energy compound. So any compound on its catabolism, if ATP is produced, that compound is also called a high energy compound. Okay. 1,3-bisphosphoglycerate to 3-phosphoglycerate. At that during the conversion, ATP is produced. So 1,3-bisphosphoglycerate contain more energy than that of ATP. So it's a high energy compound. Clear? So in that way, any reactions where ATP is produced, that's another one. Another one is phosphoryl pyruvate. So phosphoryl pyruvate is another compound. Cyclic AMP, cyclic GMP, ATP, this all can be written on the high energy compounds. Okay. Then I'm going for the next topic. Yeah. A number of questions are there on vitamins. In that, vitamin A and vitamin D are repeated essay questions. Beriberi, Pellagra are repeated 3-mark questions. Vitamin C, functions of vitamin C or scurvy, a repeated question. Gum-related problems. So dental related one, important question. Rickets, vitamin D asked the question. So several times rickets has also been asked as a short note. Vitamin K cycle is another short note. Biochemical function of vitamin B12. See, only just 2 reactions. We are studying that 2 reactions in different area. That's it. I'll just show anyway. One has already been said. Conversion of homocysteine to methionine back. There B12 is needed. Another one I'll say once again is in the breakdown of short chain fatty acid towards succinyl CoA. So that's the important question. Only 2 reactions for B12 carrying 3 marks. TPP, we have a number of reactions. TPP, thiamine paraphosphate, 2 reactions mainly. Oxidation decarboxylation and transcription reaction. PLP, functions of PLP, it's not given here. As you can add up as a 12th question. PLP, B6, just a number of functions. So these all can be very important questions anyway. Vitamin D, I'll be telling because calcitriol, active vitamin D, has action in calcium metabolism. So I'll try to discuss that at that time. Okay. So these are some important questions on vitamins. Then acid-base balance, renal regulation of acid-base balance, very important, repeated question. Blood buffer several times has been asked. Once or twice, metabolic acidosis, respiratory acidosis has been asked, or metabolic alkalosis has been asked. So write the normal values there. Okay, blood buffers, say about the bicarbonate buffer, phosphate buffer, protein buffer, hemoglobin buffer. Renal regulation, as you know, for tabular columns, where excretion of H+ ions, as free H+ ions, as ammonium ion, as titratable acid, and then 4th is reabsorption of bicarbonate. Okay, important one. Then minerals. Important questions are, of course, calcium. One repeat question is calcium. What's the normal level of blood calcium? How is it maintained? I'll be telling about that. Another short note that has been asked is about iron absorption. I'll say it's important at that time. That also I'll be explaining. Fluorosis has been asked once or twice to you. Copper in that Wilson disease has been asked once. So these are some questions that has been asked in minerals. Hemoglobin and heme metabolism. Of course, heme synthesis and porphyria is an essay. Heme catabolism is a 5-mark question. Differential diagnosis of jaundice can be a 5-mark question. Sickle cell anemia in the hemoglobinopathies. Sickle cell anemia several times has been asked. Once or twice thalassemia has been asked. Bohr effect. It doesn't affect C. That's important question that we will be studying that from in the respiratory regulation of acid-base balance also. Acid-base balance. That's about the Bohr effect. What's the Bohr effect? Hemoglobin's affinity for oxygen decreases in presence of acid or H plus ions or carbon dioxide. Every day, every time it happens in our body, when the hemoglobin carries oxygen towards the cell, their H plus ions are more because of metabolism, or carbon dioxide is more. Because of it, it will be able to unload the oxygen. Hemoglobin will be able to unload. Why? Because affinity they will uses. That's called as Bohr effect. Bohr effect means the effect of H plus ion, carbon dioxide, or acidic nature on affinity of hemoglobin for oxygen. Okay, so these all question has been asked several times. Then function test. In that liver function test, one question. As I see there, already we have about liver enzymes. We have some critically important enzymes related to liver, AST, ALT, ALP. Along that, your body have done in practical classes, total protein albumin. Only one thing is remaining is bilirubin. These are the liver function tests. Simple as that. The most simplest way I can say. Okay, then a renal function test. A repeated question is about the clearance test. Okay, it can be as commonly as clearance test or as creatinine clearance test. Nucleic acid chemistry metabolism, structure of DNA, number of times, structure of tRNA, number of times, biological importance of nucleotides, hyperuricemia of gout, Leishman syndrome. This question has been asked. In molecular biology, genetic code has been asked as a short note. Transcription, translation, there, no. They ask about the inhibitors of transcription, inhibitors of translation, inhibitors of replication has been asked. Okay. Then instrumentation, that's a surprise topic actually. We'll always go for metabolism alone, but please note that number of times Beers-Lambert's law, a colorimeter, ELISA, RIA has been asked. And the last topic is nutrition. Dietary fiber, glycemic index, BEM, once or twice the word nitrogen balance has been asked. So these are the questions that has been asked or repeated several times. So these are the important questions actually. Now I'll just say about the gluconeogenesis. As I said earlier, essay question at gluconeogenesis. Definition. Genesis means what? Synthesis. Neo means what? New. New glucosynthesis. Or so our first, if the question is gluconeogenesis, write a subheading definition. So hereafter I'll be discussing some important questions. It is just to make you aware how to write. Okay. So I'll be rushing through, but please now here at this point of time, just check the subheadings. Definition. First subheadings. Synthesis of glucose from non-carbohydrate sources. Then say what are the sources of it. Lactate, glucogenic amino acid, short-chain fatty acid, glycerol. And where it takes place, 90% liver and 10% in the renal cortex, outer part. Okay. Then next step, significance. Why you need gluconeogenesis? That's the next subheading. During starvation, whenever the glucose level is less in our body, our brain and RBC requires continuous supply. RBC, sole source of energy is glucose. So at that time, gluconeogenesis helps to maintain the blood sugar level, thereby gives energy for the RBC or the mitochondria less cells. That's the important point, actually. Pathway that maintain blood glucose during starvation. You know that glucose we get if you are on starvation, we get glucose from glycogen. But that gets depleted within around 12 to 18 hours of fasting. After that, the glucose is produced by gluconeogenesis. Then comes the major steps. Another subheading should be major steps. All the three irreversible steps of glycolysis. How can we bypass that glycolysis pathway to go back to glucose? That's what. steps which helps to overcome the irreversible steps of glycolysis. So we'll be saying three things: converts the pyruvate to phosphoryl converts the fructose 1 phosphate to fructose 6-phosphate, and glucose 6-phosphate to glucose. Okay, so the right steps, and this I'm just showing you to say that you are supposed to write all the things, not by just writing pyruvate oxalate. Instead, pyruvate, the help of ATP and carbon dioxide, and say pyruvate carboxylase, coenzyme biotin, converts to oxalate. This happens in mitochondria. Then oxalate, the remaining steps in cytoplasm of liver or in renal cortex. So it should be transported. There comes a malate aspartate shuttle. Draw that shuttle, whereby oxalate is impermeable to the mitochondrial membrane, but malate and aspartate is permeable. So converting to malate, then malate goes to the cytoplasm, then convert back to oxalate. Using the enzyme, the enzyme is malate dehydrogenase. Okay. With the enzyme name, suppose write all the things. Then how the oxalate is converted to pyruvate? Again, use of GTP, enzyme name. So write in this way, with the enzyme, the coenzyme, everything. Okay. Then comes the enzyme phosphoryl carboxykinase. Then converts the fructose 1-bis-phosphate to fructose 6-phosphate. Enzyme is fructose 1-bis-phosphatase. Last step, glucose 6-phosphate to glucose. This is enzyme glucose 6-phosphatase. So these are the four major reactions. Steps, four major reactions by which the three irreversible steps are bypassed. Okay, and then we have a shuttle, malate aspartate shuttle. Then after I did this, write about how the glucose is synthesized from different sources. Now we'll say about synthesis of glucose from lactate by a cycle called as Cori cycle. This happens during severe exercises. In anaerobic conditions, all the glucose converted to lactate. You want to prevent lactic acidosis, at the same time, you want the nozzle to be provided with the glucose. This happens with the help of a process of gluconeogenesis, and the cycle is called as Cori cycle. Then synthesis of glucose from glycerol. So when you are on fasting, slowly the fat reduces. Fat content reduces. How? In adipose tissue, the fat converts to glycerol and fatty acid. Then this glycerol converts to glycerol phosphate, then to dihydroxyacetone phosphate, and goes to the glucose. This is the way by which glucose is produced from glycerol during the starvation condition. From odd chain fatty acid. All fatty acid cannot synthesize glucose, only odd chain fatty acid can synthesize. Odd chain fatty acid on beta oxidation produces a compound called propionyl CoA. Propionyl CoA converts to methyl malonyl CoA, methyl malonyl CoA to succinyl CoA using B12. This is one reaction where B12 is utilized. Converts from methyl malonyl CoA to succinyl CoA. So two reactions of B12. One is this one, methyl malonyl CoA to succinyl CoA. Another one is conversion of homocysteine to methionine. Okay. I'm trying to say the function of B12 also. So this is another thing about source of glucose, non-carbohydrate source. And this succinyl CoA entered converts to glucose. Goes to the reversal of glycolytic pathway, goes. Or we call it as gluconeogenesis pathway. That's how the odd chain fatty acid can be utilized for synthesis of glucose. Okay, then again, during starvation, maybe the muscle protein breaks, converts to amino acid, amino acid breaks and produces ammonia. What happened to that? Glucose converted to pyruvate. This pyruvate takes up the amino acid and converts to alanine. Alanine goes to liver. In the liver, it converts back to pyruvate. removing the ammonia. Now listen, I said transport of ammonia as a subheading in urea cycle. This is one thing. From the muscle, ammonia is transported as alanine. Ammonia produced in the muscle combined with pyruvate and form alanine. This alanine comes to liver, convert back to pyruvate. Hope you understood this. Then this pyruvate through glucose pathway convert to glucose. Okay. So provide glucose of muscle. See the second one. Transport of ammonia to liver for urea synthesis. Or other word, detoxification of ammonia. So from the muscle, ammonia is transported as alanine. From the brain, it is transported as glutamine. Please not. I'm telling about urea cycling. Okay, now comes back. Other glucogenic amino acids. Glucogenic amino acid, on its breakdown, produce some compound that are present in the thesis cycle, intermediate the thesis cycle. And then convert back to glucose. I'll show some examples. Glutamic acid, arginine, proline, histidine, they all break down, produce alpha-ketoglutarate. Alpha-ketoglutarate converts succinyl-CoA, succinyl-CoA fumarate, fumarate to marine, marine to oclastate, then oclastate through marine transfer shuttle goes out, convert to phosphoryl-pyruvate, then to glucose. Goes like that. Valine isoleucine, methionine, or odd-chain fatty acid all produce succinyl-CoA. Succinyl-CoA convert to fumarate, fumarate to marine, marine to oclastate, then goes out through marine transfer shuttle. It goes like that. So our point is, this is the point. And phenylalanine tyrosine break down the last process fumarate. Fumarate, it goes like that. So this is what the point is. On it breaking down, produce intermediate the thesis cycle or pyruvate. They are and can be used for glucose synthesis. So this is what. That's all about. So these are the points. And then one more point is suppose, right? How the process is regulated. Regulation of gluconeogenesis. There is suppose like the hormone regulation and allosteric regulations. Okay. So these much subheading is about gluconeogenesis as an essay. Definition, sources, site, major use of gluconeogenesis or significance of gluconeogenesis, major reactions, and the major from the major sources from which glucose is synthesized. Okay, that's about gluconeogenesis. So in this way, write an essay. Now about a short note. Important short note always comes is HMB pathway, significance of HMB pathway. A 3-mark question comes. See, if you're able to put in schematically as an examiner, I'll be happy. Because very easy to correct. One glance, I can see the importance. There are two types of significance, biochemical and clinical. Biochemical significance, see, either NADP is produced, pentose phosphate. Say the use of it. Clinical importance, if the G6PD is there in the enzyme HMB pathway, that can lead to drug-induced hemolysis, methemoglobinemia. See, if you do this and write some points, no doubt, easily you can fetch the marks. So in a digital evaluation process, schematic representation means a lot. The anatomy, you draw the diagram and say the points, The same way, physiology, biochemistry and all, schematic representations means a lot. For us, very easy to correct the paper and put the marks. We'll see the use of NADP. We have a lot of NADP use for NADP, at least 5. Can enlist one by one. Any lipid synthesis, you need NADP. So, some significant HMB pathway. Now, listen. For this one, HMB pathway, we know that glucose is mainly for energy, right? But this pathway is not for energy, for something else. Or other words, significance of HMB pathway means the question is, other than energy, what is the use of glucose? Now listen, we said we produce NADPH. Thereby, glucose can be involved in lipid synthesis. Hope it's clear. Please understand that. If you understand that way, you won't forget. Don't by heart. We know that glucose is for energy, but here, this is not only for energy, for something else also. What is that? NADPH pentose. Thereby, what's the use? NADPH. Thereby, glucose is indirectly for lipid biosynthesis. Free radicals scavenging. This step, I'll show once again. In the RBC, this O2 superoxide converts to H2O2. Both are highly reactive. Then use glutathione, converts to water. Then glutathione become in oxidized form. It is going back with the help of NADPH. This is the reaction to say that free radical scavenging effect. This diagram is schematic representation is more than enough, but provided write the name of enzyme. GPX means glutathione peroxidase. GR, which stands for glutathione reductase. GSH is glutathione in the reduced form. GSSG is glutathione in the oxidized form. Okay, one more point, please note. I said two major compounds are NADPH and pentose. See, we have one more compound NADH. If anybody write the word NADH, gone. You won't get marks. Because NADH is for energy. This is not for energy purpose at all. NADPH is different. NAD is different. So please note HMP. So NADPHP. Study like that. And pentose. Again, one more P. Pentose phosphate. Okay. So HMP pathway. Third, that by scavenging free radicals, it can preserve the integrity of RBC membrane, prevents methemoglobinemia, prevents cataract, or maintain the transparency of islands. We can't say prevents cataract, but it can prolong cataract genesis. Or maintain, this is the better word, maintain the transparency of islands. Bactericidal action in macrophages. That's also with the help of NADPH. And pentose phosphate for nucleic acid synthesis, purine pyrimidine synthesis. Okay. So that are the other uses of NADPH. And one more for NADPH for the detoxification of some drugs, some compounds, NADPH is essential. So write that. These are the 7 uses of NADPH. You can enlist that. Okay. I'll maybe 1 or 2 more topics I'll discuss today because of the constriction of the time. Another one question is about the iron absorption. Why iron absorption is important? Because listen very carefully. Most of the biomolecule by compound what we take, it level the body is maintained at the level of excretion. That means if we take, okay, it's absorbed, extra are excreted out. For example, vitamin B tablet, if we take B complex tablet, next day morning, your urine sample will be yellowish in color. Why? Because the extra B complex, they are soluble in water, so just get that. But iron, that's the difference. Iron level the body is controlled at the step of absorption. If you need, it is absorbed. If it is not, it is not absorbed. If it is not, it is not absorbed. That's the thing. So it's important. And this process tightly regulated. So whenever the iron absorption is asked, they're asking about one more thing, a theory to say about the regulation. We call it as mucosal block theory. If the mucosal block theory is asked, write about iron absorption also. Iron absorption I'll put very in a simpler form. I'll just show you that. Okay, what are the only the first form is iron, vitamin C is essential for it. Only ferric, okay. Ferric reductase convert that. Some compounds inhibited. I'll just show them a picture. See, in this way, intestinal lumen, intestinal cell, blood, and other cells. Our daily requirement is around RDS 20 milligram. Our green leafy vegetables contain, dates contain, pomegranate contain iron. Jaggery contains iron. But that iron will be ferric form, Fe3 plus. So put a circle right in that Fe3 plus, not red color. That's what it may be in the paper. Then these ferric ions cannot be absorbed. These ferric ions are converted to ferrous ion, Fe2 plus form, with the help of reduction of Fe3 plus to Fe2 plus. So the enzyme ferric reductase. Vitamin C. Of course, we will study vitamin C, function of vitamin C. Helps in absorption of iron. Convert to ferrous ion. Then this ferrous ion is a Fe2 plus, so divalent. Divalent metal transporter, DMT1. Through that, it is transported in or it is taken up by the cells. Then either convert back to Fe3 plus with the help of peroxidase and stores in the cells as ferritin. Or as per the need, if the blood contains a lot of transferring ion transporting fellow, then this ferrous form is exported. So that for that we need a protein transporter called as ferroportin. The yellow color one is ferroportin, it's a transporter. It's a membrane, okay? This is a membrane. That's why I put like that. It's a transporter here. Ferroportin. Here is DMT1 ferroportin. Then it enters to the blood. Converts to ferric ions. Enters to transferrin. Transferrin with the help of receptors goes to cells. This ion is stored as ferritin. So this diagram shows about the storage from ferritin, transport from transferrin to transfer the DMT1, ferroportin. For that iron, vitamin C is required. The enzymes involved, what's the RDA, what's about the sources, everything is there in one paper. A small diagram. Don't you think this will be easily get you marks for that? Of course. If you put this thing in, if you want to write, you write. I tell you, most of the examiners will just see this thing, happy, leave it. Maybe some examiners will go through and read. Okay, if you want to write, you write. But this, all the points are there. Very easy to fetch the marks. Very easy for the examiner to correct this also. And then, after this, don't stop here. My point is, don't stop here. Whenever the ion absorption is asked, say about how the process is controlled. So this is the luminal part. That means that the pathway through which the food goes. Luminal part, mucosal cell, basolateral side. That means that the blood cells. Fine. Okay. And the level of this DMT1 and ferroportin is controlled by another protein produced from liver. Okay. It's called hepcidin. Liver. Just understand that way. Hepcidin controls the level of DMT1 and ferroportin. Whenever the iron level is high, this will be high and it will inhibit the production of DMT1 and ferroportin. So iron will not be absorbed. Clear? If the iron level is high in the body, hepcidin will be high and that will inhibit the production of DMT1 and ferroportin. Thus, the absorption is controlled. Okay, there comes the word mucosal block theory. This is the point. As I said earlier, iron absorption is unique because iron level in the body is maintained at the level of absorption, not by excretion. When the iron stores are depleted, absorption is more. The quantity is iron, enough amount is there, absorption or sufficient quantity is stored, then absorption is decreased. How? Mainly 4. Level of divalent transporter, DMT1 and ferroportin are controlled by hepcidin. Then from ferroportin to blood, for that a lot of transfer should be there in the blood. Movement of iron across the mucosal cell through ferroportin depends on the availability of iron. Whenever the anemia comes, third important, anemia comes, erythropoietin will produce, that will increase absorption of iron. 4th, as I said earlier, some amount of iron will be stored in the mucosal cells. During the desquamation process, through the feces that go out. So temporary storage mucosal cell is lost during desquamation. In this way, ion absorption is totally controlled. That presentation is also important. And my point was two things. One, try to put the points in simpler way, schematic way. Along with that, some extra ideas there. Ion absorption will not stop with just ion absorption alone. You should write about mucosal block theory. And if the question is mucosal block theory, write about ion also. Okay, another important question from our mineral metabolism is calcium. But just need that. Okay, before that, you can write schematically also. Whenever the anemia comes, there will be increased secretion of erythropoietin. Erythropoietin decreases the production of hepcidin. So, increase. So this is what hepcidin, and they are inversely proportional, like or indirect. Decrease production of hepcidin, so increase production of DMT and ferroportin, and this increase absorption. And then whenever the iron level is more, hepcidin is more, that will reduce the level of ferroportin and DMT1. Thus, iron absorption decrease. This is what I meant. Okay. Now I will go to an important question: calcium. Many a time the question has been asked, what's the normal level of blood calcium? How is it maintained? There, one important portion the students miss. Of course, the normal level, you can write the word 9 to 11 or 8.5 to 10.5. Anything. Either you can write 8.5 to 10.5, 8.5 to 10.5 milligram per deciliter, or 9 to 11, easy one. 9 to 11 milligram per deciliter. Now, whenever the blood normal is asked, we are asking something else also. In the blood, calcium exists in three forms. One is ionized form. That's the only one which is functionally active, metabolically active. That you know or not. Because of metabolically active ionized calcium level changes in several clinical conditions. We should be aware of that. That is why this question is important. These points are important. Calcium exists in three forms. One is ionized calcium, around 50% is ionized form. Second is complex form with a citrate, it is attached with citrate, phosphate, bicarbonate. And another more important, that's only 1 milligram. Another around 4 milligram bound to protein. Okay, these two are diffusible. Some cells can diffuse out. And last one is protein bound calcium. For the blood, it cannot go, it's a non-diffusible form. So this should be written. Always calcium in the blood. Normal level of blood calcium now is maintained. Normal blood calcium level, and writing these things. This will be these 3 points will carry a specific marks for me. Please note that. And then say, how is it maintained? This word. At least you should enlist it. We have hormonal and non-hormonal factors. Please note that. Now, hormonal and non-hormonal factors. Hormonal calcitriol, PTH and calcitonin. Non-hormonal factors are there. As I said, acidosis, alkalosis, serum albumin, any proteinuria condition decreases. Children will be more. And calcium level depends on the phosphorus level. Important. Renal threshold is around 10. So these are the non-hormonal factors. Write that and explain as per the question marks. Okay, marks for the question. If it's an essay, write totally. Explain on everything. If it is an 5-mark question, you can explain on hormonal factors, but just list out the non-hormonal factors. Okay, I hope I have gone through some important topics in this one. Just went through all the questions and some very important questions, some important topics. My idea by going through the important question was just to tell you how to write and what are some points what you are usually the students misses that should be incorporated, which can fetch you is good marks. See, after all, writing an answer sheet, thereby you are trying to impress the examiner. When you write some extra points or points from different dimensions, of course, the examiner will get influenced by that, will get impressed by that. That's the point. So write accordingly. Try to impress examiners by writing the different dimensions of a question. Hope you understood this topic. Hope this was useful for you. And thank you for your patient listening and all the best. All the best for you. My prayers and wishes for you for a better, best results. Thank you.